Well, uh, thanks to all of you for joining this community webinar. We're all trying to cope with the difficulties of the coronavirus pandemic, and in part a consequence of years of failure of the government to prepare the nation. In fact, given the efforts of the Trump administration to downgrade and undermine the nation's scientific resources, it seemed appropriate to start with an introduction to the actual etiological agent of the disease itself, the coronavirus. So this slide talk will be in three parts. First, I'll talk about the structure of the actual virus particle itself. Then we'll go into the development of vaccines and therapies. And then I'll end up talking about federal budget choices. So first, it's important. Many people are confused by the difference between bacteria and viruses. Diseases caused by bacteria, familiar to most of you: cholera, pneumonia, diphtheria, tuberculosis. Hopefully you hadn't had them. Whooping cough, urinary tract infections, meningitis. And then diseases caused by viruses. They include influenza, polio, HIV/AIDS, head colds, cold sores, smallpox, chickenpox, warts. Now, viruses are parasites. They can't reproduce by themselves out in the environment. They have to find a particular host cell, they infect the cell, and then inside the cell, they take over the cell's reproductive machinery and direct it to produce new viruses. And then the new viruses are released. Often the infected cells die. The new viruses are released. They find another cell, infect again, and the cycle repeats. Now, in the United States, the history of our understanding of viruses really starts with the tragic epidemics of poliomyelitis, infantile paralysis, in the 1930s and 1940s, where tens of thousands of primarily young children were paralyzed, but also a very popular president, FDR, was afflicted and paralyzed from the waist down. And he was paralyzed all through his presidential career and worked hard not to show it, but he couldn't stand without crutches and was really seriously afflicted. The second thing that's in U.S. history is very important is the end of World War II, when millions of GIs returned home. They weren't used to paying for health care on the battlefield, just called for a medic, and then they came home to a privatized healthcare system. And in the period after World War II, the issue of healthcare, national health insurance, national health service was really a hot issue, just like it is now, the debate for Medicare for all. And in fact, I can remember as a young boy, I used to listen to between two radio programs between 5:30 and 6, The Lone Ranger and The Shadow. And in the break between, in those days, you'd often hear advertisements warning against the danger of socialized medicine. And in fact, the proponents of keeping healthcare privatized held sway, but Truman arranged a compromise, and that was federal support for biomedical research, not for healthcare, but for research. And the then fledgling National Institutes of Health was given the kind of portfolio for biomedical research, and that has grown ever since. Now, the National Institutes of Health is a physical place in Bethesda, Maryland, but really it's an agency that distributes research funds from your taxes, taxpayers' dollars, to scientists like myself, my students, in colleges and universities and medical schools all across the country. Now, the first viruses that we came to understand were viruses that infect plants. Plants get diseases, and this is an electron micrograph on the left of a tobacco mosaic virus. You see it's a long rod, and on the right is its underlying structure. There's two components. In the center, spiraling around, is the long, thin RNA molecule that is the genetic material. It controls the heredity and the reproduction, eventual reproduction of the virus. And it's protected by these kidney-shaped blobs. And each of those is a protein. They're all identical. They're called coat proteins or capsid proteins because they form the coat and capsid of the virus. These tobacco plants, the leaves, you see brown splotches on the leaves. It's very obvious, and there was great loss to the tobacco crop in the 20s and 30s. The second breakthrough was discovering the nature of viruses that infect bacteria. Yep, bacteria get viral infections too. And viruses that infect bacteria are called bacteriophage, from the French to eat bacteria. Here's an electron micrograph of bacteriophages that infect Salmonella typhimurium, often associated with food poisoning in humans. And you can see it has it's round, but it has a little tail. It's rather distinctive. Now, it turns out that, here's an electron micrograph. There's a phage particle about an hour before this sample was prepared. This particle attached to the surface of the bacterial cell, it's the cell membrane, injected its DNA into the cell. It's white, it's empty because it's injected its DNA into the cell. And inside the cell in the hour that followed, hundreds of new virus particles were formed. This cell will break open and would have broken open in 20 minutes and released all these virus particles, which would infect other cells. It was the study of these bacteriophages that led to this understanding that DNA was the genetic material, that DNA encoded RNA molecules that were messengers, and they specified the structure of proteins that are the enzymes and building blocks of all cells. You don't have to understand, it's enormously complicated how this works out, but just remember there's the blueprints, which can be DNA and RNA, and then there's the actual machinery and building blocks, which are the proteins. Now, it turns out that most of the viruses that we're concerned with are round or spherical, and in fact, they actually are icosahedral. Now, most people watching this, it's a long time since you took high school geometry, but you might remember that there were the regular Euclidean solids like tetrahedrons. Most viruses are icosahedrons. Icosahedrons have 20 faces and 12 vertices. The vertices are kind of the corners. And these are viruses. The big one on the left is herpes that causes cold sores. One of the viruses causes infantile diarrhea. A number of these are plant viruses. Among the smaller viruses are poliovirus, polyomaviruses. The coronaviruses turn out to be round too, not quite spherical, but close to it. So here's an electron micrograph of adenoviruses. These are viruses that infect your upper respiratory tract and are a cause of colds and coughs and head colds. And I'm going to show you, and there's a structure at much higher resolution that uses the modern technology of cryo electron microscopy. These blobs are the proteins that are forming the shell of the virus. But you'll see at the vertices, there are these skinny spikes. Well, these spikes may not look like much, but they're enormously important. It's through these spikes that adenovirus attaches itself to the cells of your upper respiratory tract. So they're important for infection. And it also turns out that the way your body fights this virus is it makes antibodies. We'll talk about antibodies a little later on. And these antibodies they glomp onto those spike proteins, and that prevents the virus from infecting. Coronavirus has spikes too, and they are just as important for coronavirus as adenovirus. Now here's influenza virus. Influenza virus has an additional feature around the protein coat or capsid. There's a membrane, like the membrane around cells. It's actually derived from the cells in which these virus particles were made. And sticking out of these membranes are these spikes that are very important. The influenza virus binds to receptors on the cell surface. That's the surface of the cell. The cell decides it looks like it's something it might be able to use, and it invaginates and brings the virus inside the cell. The kind of virus tricks the cell in a way. And then the cell breaks down the virus particle, but that ends up releasing the genetic material, which for influenza virus is RNA. And that releases the RNA into the cell, and then a complex process ensues where those molecules are replicated, new proteins are made, new virus particles are made, and they're released from the cells. So that brings us to coronavirus. By the way, the name just refers to this appearance of a corona around the viruses from the spike protein sticking out. This has RNA as the genetic material. The reason it's, for this talk, the reason you have to know the difference between RNA and DNA is because the replication of those molecules is a little different. And an important class of antiviral drugs are those that interfere with the replication of the genetic material. But the ones that interfere with DNA don't work on RNA and vice versa. This is a very common virus among mammals, birds, typically causing mild respiratory illness, probably pretty common in the human population. Not this particular one that's caused this outbreak, but its cousins. Two major serious outbreaks of a related virus were the SARS severe acute respiratory syndrome in 2002, 3, and 4 in Hong Kong and Asia. And then 10 years after that, the MERS virus, Middle East Respiratory Syndrome, 2012, 13, 14 in the Middle East. The current outbreak, the disease, coronavirus disease 2019, that's the name of the disease, COVID-19. The actual virus name is SARS coronavirus 2. Now, here's what it looks like at higher resolution. It's not colored. The color has been added to make it easier to look at. And those red things sticking out, those are the spike proteins. Those are the proteins that coronavirus uses to bind to your respiratory and lung cells. And it's also, we hope, would be the target of antibodies, of your body's antibodies, the antibodies that your white cells make that will inactivate the virus, which again, we'll talk about in a little bit. Now, so outside of the spike proteins, inside is the RNA, and the RNA is coated with a set of proteins called nucleocapsid proteins, and there's some additional proteins in the membrane that are important for the virus getting into the cell and getting out of the cell. But if you just understand that these spikes are key to get into the cell, you're in good shape. Now, people want to know, well, what kills these viruses? Everything kills it. 70% ethanol, 90% isopropanol. That's the rubbing alcohol that you buy in a pharmaceutical, in a drugstore. Dilute bleach, like you put in your washing machine. Soaps and detergents. Soaps and detergents not only clean your hands, they actually kill the virus. Ultraviolet irradiation, either from a UV lamp, but full sunlight. If you're walking out in the full sunlight in the daytime, virus particles are inactivated. The heat of cooking and drying, right? These particles have to stay wet to stay active. I'm not going to spend a great deal of time in a debate about how long the aerosol droplets stay in the air. But the point is that when they dry, the virus is essentially inactivated. So it is transmitted when you sneeze, when an affected person sneezes or coughs or laughs or yells. And so there's small droplets. Big droplets fall to the ground. You don't have to worry about them. Small droplets stay in the air longer. But on the other hand, they also dry out faster because they're small. So your nose reduces the risk of virus particles getting to your throat. A mask reduces the risk of the virus particles getting to your nose. Social distancing reduces the risk of them getting to your mask. And together with hand washing, these countermeasures work very well, right? And so if we all wear masks when we're outside, if we all wash our hands, we can limit very, very much limit the transmission of viruses from one person to another. On the other hand, if 5 people are partying together and laughing and yelling back and forth and hugging and kissing each other, and one of them is infected, the other people are at very serious risk. Now, how does your body fight the virus? How do you get over it? So we all have in our body, all of you know about red blood cells because you've bled and you've seen the red color, which is due to the red blood cells. And we also have in our bloodstream and also in our lymph white cells, 2 classes of white cells, B cells and T cells. And these white cells, they're also made, by the way, in the bone marrow. They're cousins of red blood cells. But one class of them is very important. One class of B cells synthesizes a group of proteins called antibodies. These are Y-shaped proteins, the blue and green molecule on the left. There's actually 4 protein chains stuck together. But these antibodies are able to recognize, can't do that, are able to recognize foreign objects. They recognize viruses, bacteria, parasites, pollen, toxic agents, tumor cells, which are making proteins that they shouldn't. So in blue, you see an antibody. It's binding to proteins on the surface of the cell in orange. We use the term antigen. Antigen is what the antibodies recognize. Very confusing. Antibodies do the damage. Antigens are the target. So here you see 2 antibodies, A and B, are binding to antigens on the cell surface. Antibody C there is binding to antigens that are floating around, let's say, in the bloodstream. Now, you have in your your blood, all of us have millions and millions of white cells, billions of white cells, and they are making a great variety of slightly different antibodies. So, for example, I've had the flu vaccine, and so my body has been stimulated to make antibodies against flu. I actually had polio when I was 3 years old. In my legs, when I was young, I had to wear special shoes and braces. I was very lucky. I recovered, but my youth was marked by the disease, absolutely. But this was before the Salk vaccine. In fact, I have, probably carry to this day antibodies that recognize polio and would protect me. Most of you have been inoculated against mumps, measles, smallpox. So you have in you white blood cells that are making antibodies that recognize these viruses and zap them. Now, what's a vaccine? A vaccine, typically, for example, on the left of this picture, is a form of the virus that's not very infectious. It can be damaged by radiation, by chemicals, by heat, so it itself can't cause a serious infection. But it's recognized by your white blood cells. The white blood cells that recognize it are stimulated, and they start turning out antibodies that will bind to the vaccine virus particles and clear them from the bloodstream. Those white cells, it takes days and weeks for them to build up to high concentration, but normally they hang around for months to years. And so after you've been vaccinated, you have white cells that make antibodies that will zap viruses getting into you, and the protection can often last for years. So there's a very active effort going on everywhere in the world, biomedical labs all across the country, biotechnology companies, pharmaceutical companies, to make vaccines using coronaviruses that will stimulate your immune system. Now, with modern technology, we can make vaccines that don't depend on the whole virus, but depend on the isolated spike subunits. Those are those funny shaped blobs in the lower right of this image. If you inject a person with the spike proteins of a virus, and that's what's done with the flu vaccine, your white cells respond, make antibodies against the spike proteins, And then if a real virus shows up, they zap the real virus. Now, vaccines have been enormously effective against many, many, many diseases, but it takes time to develop to develop vaccines. Think about it. First, you got to make sure that the vaccine itself isn't going to make you sick. So you have to have what's called a phase 1 trial where a small number of people are exposed to the vaccine, and you have to watch them for weeks and maybe months and make sure they don't get sick. Once you know that they don't get sick from the vaccine, then you have to find out, does the vaccine induce an immune response? Does it stimulate your white blood cells so that you'd be protected from the actual infectious virus? Then you might do a phase 2 study with a smaller number of people to see, is this vaccine effective? But of course, in this society, you have to make sure the vaccine works in young people, in old people, in men and women. And you need a trial with a large number of people. And these have to be people who are going to be challenged after they get the vaccine. They have to agree to be infected by the actual virus. In the old days, this was done with prisoners who didn't have much choice, or in third world countries. Nowadays, we have stronger regulation of that, and we get volunteers, but it takes time. It takes time to develop a vaccine. A little quicker, but not super quicker, is the possibility of developing drugs. So this is a complex diagram, shows on the left the virus entering into cells and then releasing the nucleic acid. And then the cell has to make the proteins of the virus, and they have to build a new virus particle. I spent most of my professional life studying these steps, the intercellular assembly of the virus particle from its constituent proteins. And there's a number of places in these process where it's possible to intervene. So there are drugs that interfere with the reproduction, the replication of the RNA of the virus. There are drugs they would develop with HIV that interfere with the assembly of the particle. There's also some drugs that interfere with that very first step shown on the left, where the virus spike protein has to bind to the receptor. So these are, in development all across the country and around the world. Now, the research that supports the development of vaccines and drugs is not funded by Medicare. It's not funded by Medicaid. It's not funded by private health insurance. It's not funded by hospitals. It's funded almost entirely by the National Institutes of Health, National Science Foundation, Center for Disease Control, and a few other agencies. These agencies get their budgets from the Congress, from our tax dollars, and then they distribute them around the country. In the Boston area, almost all of the young scientists, graduate students, research technicians, postdoctoral fellows who are working on these kind of problems at Harvard, at MIT, at Tufts, at Boston University, Boston College, UMass Boston, they are all paid by the federal government. MIT graduate students are not paid by MIT. They're paid by you, by your tax dollars, but not from health insurance through these appropriations, the NSF, NIH, CDC, etc. So those budgets are enormously important. And it's not widely known that our response to these diseases absolutely depends on this pool of biomedical researchers and is outside the healthcare system. Now, almost all of the information that I've showed you, those electron micrographs, all of the stuff about the spike proteins and antibodies and white cells, all of that is information that came from biomedical research carried out after World War II in laboratories across the nation. That's absolutely critical for our ability to respond to these kind of new threats. Unfortunately, the federal budget over the last decade, I would say, has been really, really badly distorted. So here, this pie chart shows how Congress voted your tax dollars in 2017. It's called the discretionary budget. It doesn't include Social Security. That's a trust fund, not paid from income tax dollars. Doesn't include Medicare. That's a trust fund, not paid by income tax dollars. This is all your income tax dollars. It was about 1.2 trillion that year. The blue is your tax dollars that went to the Pentagon for foreign wars, preparing foreign wars, new nuclear weapons, buying and maintaining old weapons, maintaining 800 bases abroad. The NIH slice is up there in brown, 34 billion. It was less than 3% of the total congressional budget in 2017. That's about still true this year. So this is the money that the nation allocates to deal with all of the diseases that we suffer from: heart disease, stroke, Alzheimer's, diabetes, viral infections, bacterial infections, antibiotic-resistant infections. All of the resources that we devote to protecting our health is 3% of that federal budget. How should we compare the security we get from the $700 billion military expenditure and the $34 billion NIH expenditure? Let me give a quick example. I'm coming to the end. Alzheimer's. Everybody, most people listening to this slideshow know somebody who's suffering from Alzheimer's. 5 million Americans at present. Cost the healthcare system about $260 billion, about 20% of the Medicare budget, just taking care of those millions of people suffering from Alzheimer's. The National Institutes of Health, now the best healthcare in the world won't help you right now if you get Alzheimer's. We are totally dependent on biomedical researchers and pharmaceutical researchers coming up with a way of preventing or alleviating or curing the disease. Well, the NIH budget for Alzheimer's is about $1 billion. So we have a problem that costs the nation $260 billion. We're spending $1 billion to tackle it. And of course, the human toll, right, the suffering and the loss of productivity and the weight on the caregivers is enormous. It's crazy that only a few $1 billion are spent on that. By comparison, let's look at our nuclear submarines. The government is proposing to buy. We have 14 of these, each armed with 24 Trident D5 missiles with 12 nuclear warheads each. If one of those submarines, just one, fires off all its missiles, that's enough to obliterate every major city of any country on Earth, Russia or China or India. Again, we have 14. The government is proposing to buy 10 more. It's not really the government that's proposing it. It's the military-industrial complex who gets rich off these weapons that's proposing to build them. If we got rid of one nuclear submarine, we could take care of all the shortages of masks and gowns and ventilators, in the nation. It's billions of dollars going to waste. And that's what needs to happen. We're not going to be able to solve this coronavirus crisis without investing an enormous increase in sums, not only biomedical research, but of course the actual healthcare system and getting healthcare for all and Medicare for all. So I'm going to close here. These are some sites where you can get more information about these points. I want to thank you very much for listening. You can contact me at keepcambridgelivable@gmail.com, or this slideshow will be on the Cambridge Residents Alliance website, also the Mass Peace Action website. So carry on, stay well, we all stick together, keep our masks on, stay socially distanced, and we'll get out of it, and hopefully we'll make a better world when we come back. Thanks a lot.